Hi, when we immobilize an enzyme into a solid support, our main objective is the catalytic reuse of the enzyme. We can either use reversible or irreversible method for the process, and reversible methods used to affect less in terms of the enzyme activity and enzyme conformation, but most of the irreversible methods are affecting the enzyme activity. But we cannot say that it will always positively or negatively affect the enzyme activity. Some of the modifications are actually good. They will increase the enzyme activity. But some other modification may be decrease the enzyme activity. So the effect of enzyme activity, the effect of immobilization on the enzyme activity vary depending on the type of enzyme and type of process, type of support. So properties of immobilized enzyme can be positive or negative. Sometimes it will increase the stability of the enzyme, but in some other cases it will reduce the stability of the enzyme. So it is, in a general sense, it is unpredictable. It can go in both ways. But generally, immobilized enzymes has several properties. First one is alteration in catalytic activity and thermal stability. So if an enzyme is immobilized, there will be changes in catalytic activity. It may either go up or go down, depending on the type process. And thermal stability, how it resists the temperature. It will also increase or decrease. But in general sense, catalytic activity will increase during immobilization. Thermal stability also increase during immobilization. But if you are doing some cross-linking or covalent binding, catalytic activity may go down instead of going up. But generally, thermal stability is always going up when we are immobilizing the enzyme in a solid support. This can be due to immobilization or by the influence of microenvironment. So if we are making the crystals of the enzyme as it is in a crystal lattice structure, it will become more resistant to temperature, pH and everything. Or it may be due to the microenvironment. If you are encapsulating in a small space, the chance of degeneration by autolysis and other compounds is less. And immobilized enzyme may have the changes in the three-dimensional structure because of the steric hindrance or attachment to a solid support or cross-linking. The three-dimensional structure may vary. And generally, immobilization improves the operational stability. Stability of enzyme generally increases during immobilization. Okay, and major problem is like steric restriction due to the confinement of enzyme into a solid support. Another immobilization method is the immobilization of the cells. Instead of immobilizing enzyme, we can actually immobilize cells. The advantage of this method is like if you want to immobilize the enzyme, first we need to grow the cell, then actually isolation and purification of the enzyme, and this purified enzyme is actually immobilized. We can actually bypass this purification step by immobilization in this cell. The cell which is making the enzyme will be immobilized in a solid support medium. Advantage of this method is like once we immobilize the cell, it doesn't actually grow. If it is properly immobilized, the cost for the growth is very less. So we don't have to provide more nutrients for the immobilized cells than the free cells. Free cells always need more oxygen. It will die quickly. It will reproduce quickly. It needs constant demand of for its metabolism. But if it is an immobilized cell, we can actually immobilize live or dead cells. So whatever type of cells we are doing, the cost of nutrients is very less in case of immobilized cells. Either we can immobilize the isolated enzyme or whole cell or cellular organelles like chloroplast or mitochondria can be immobilized. If our enzyme is situated in the mitochondria, we can actually isolate the mitochondria and immobilize mitochondria. Whole cell immobilization, immobilized either in viable or non-viable form. It's avoid enzyme purification. It preserves enzyme in the natural environment. That's a very good advantage, because when we isolate the microorganism, especially if it is an intracellular enzyme, what happens is like when we put in extracellular environment, it will go and denature. But if the enzyme is situated inside the cell, it will be more protected than the outside world. So its activity and reusability will increase. It is protecting enzymes from inactivation because the exposed enzyme may get inactivated fast, and it can be used as a multi-purpose catalyst. If you want to immobilize enzymes for a metabolic process, we need to immobilize all enzymes to get the product. But if you are immobilizing a cell is also having all enzyme. So we don't need to isolate separate enzyme and immobilize separate enzyme. It can be done in a simple step. Immobilized viable cells are called controlled catalytic biomass. Limitation of the immobilized cells, including the free diffusion of the substrate and product, is limited because of the presence of cell wall and cell membrane. So if our reactants or product is not able to cross the cell membrane and cell wall easily, it leads to the diffusion limitations and reduced productivity. And another disadvantage is like unwanted side reactions. As the cell is having different types of enzyme, it may convert the reactants into some other side products and byproducts. So that's also a limitation of the immobilized cell. So the permeability limitation can be overcome by permeabilized cells. Instead of using the living cells or the dead cell, we are using permeabilized cells. These cells have more permeability to the reactants and products. So the background is like most of the enzymes we are used in industry is extracellular in nature, but 90% of enzyme produced by the microorganism is intracellular, inside the cell. Once we isolate the cell, enzyme from the inside, it is very sensitive to the external environment. So we want the enzyme should sit in the cell, inside the cell, and we will make it more permeable. The cell will be more permeable to the reactants and the products. So in a permeabilized cell, the permeability of the cell is high. The cell wall is more permeable, or we strip out of the cell wall, or the cell membrane will be more permeable. So that the free diffusion of substrate and product is possible. Minimize the unwanted side reactions by specifically inhibiting other enzyme. They are non-viable and non-growing cells, so you don't need to provide nutrients for this cell. Very economical source of intracellular enzyme. When we are comparing the fermentation process and enzyme immobilization, permeabilized cells are more economical and used for the hydrolysis, isomerization, and oxidation reactions. Permeabilized Culvinomyces fragilis is used for the conversion of lactose to glucose and galactose. So it's actually working. Applications of immobilized enzyme. There are so many applications in immobilized enzyme. Biomedical applications, we can actually use it for treatment of inborn metabolic deficiency like phenylketonuria, or it can be used in diagnosis. You may have seen the machine used for detecting the blood glucose level. We will make a small pinch in our finger and put a drop of blood in that machine in a strip. You remember? In that strip actually containing immobilized glucose oxidase enzyme. So this immobilized glucose oxidase enzyme convert this glucose into other product, and which is detected by the machine. So the AquaCheck type of glucose detectors is working on the working with the immobilized enzyme. Food industry, it is used, as I said, the example glucose isomerase, which is used for the production of high fructose corn syrup. It's an application. Biodiesel production, we can use the wastewater treatment, textile industry, or detergent industry. All these industries are using immobilized enzyme. And if we want some examples, immobilized enzymes are used for the amino acid production. The enzyme is called amino acylase. It is isolated from Aspergillus oryzae, and it is immobilized in DAE cephadax, and its half-life is 65 days at 50 degrees Celsius. So we can actually use it for 65 days by losing only half of its activity. Actually, this enzyme catalyzing a reaction like this, N-acyl DL amino acid is converted to L amino acid, which can be used in food industry, pharmaceutical industry. It's more economical, more efficient substitute, reduction in the cost of enzyme and labor because we are using again and again. Another application is for the L-aspartic acid production. L-aspartic acid is the first industrial application of the immobilized microbial cells. Actually, in this case, we are immobilizing these cells. It is used in food and pharmaceutical industry. You might have heard about the aspartame. Aspartame is the low caloric sweetness which is used by the diabetic patients. So, which is actually synthesized by using L-aspartic acid. So, the enzyme responsible for this production is called aspartase, which is isolated from Escherichia coli. and the reaction catalyzed by this aspartate goes like this. Fumaric acid is actually converted into L-aspartic acid. You see, the operation half-life of this reaction is 680 days at 37 degrees Celsius. So we can actually use it for almost 2 years. The same thing, you don't need to feed the cell or you don't need to do anything, so you will get the product continuously. After the half-life is over, we can actually regenerate the support and add more enzyme. So we don't lose anything. Thank you so much.